Good afternoon to you all. It is a pleasure to see so many of you present at the launch of this third Global Food Regulatory Science Symposium that we are organizing within the 18th Dubai International Food Safety Conference. My name is Samuel Godefroy. I'm a professor of food risk analysis and regulatory policies at Laval University in Quebec City in Canada, the province of Quebec. And I have the privilege also to be the president and the chief operating officer of the Global Food Regulatory Science Society. It's a great pleasure for me to welcome you all on behalf of G-Force and also to thank, in fact, the organizers of the Dubai International Food Safety Conference to have enabled us to renew the tradition that we have started in 2021 by organizing within the Dubai International Food Safety Conference a Global Food Regulatory Science Symposium. This year's symposium will include a number of sessions, in fact, exactly 4 sessions that are dedicated to food regulatory science, various facets of food regulatory science. Our events, our sessions will be enabled in a hybrid mode. So I would like also to welcome those colleagues who are joining us online right now, but also those who are going to be either watching us live on the YouTube channel of G-Force or subsequently. I mentioned that we will have 4 sessions. The first session will be this afternoon, and it is dedicated to one of the activities of the Arab section of AOAC International related to the performance of gluten methodologies. Tomorrow, we have a session in the morning related to risk assessment, and we're going to be examining current and emerging issues in food risk assessment with a focus on the Arab region, but also at the global level. And the third session is going to be also tomorrow morning at 11 a.m. on harmonization of food regulatory frameworks. And then we will have a fourth event in the form of a workshop that is co-organized with the World Health Organization on data-based decision-making processes, mainly working on efforts to make available data to support the Arab region make risk-based and science-based decisions as part of their regulatory frameworks. So as I mentioned, all these 4 sessions will be managed in a hybrid format and will be broadcast live as well on our YouTube channel. I'm also privileged here to welcome the colleagues from the Arab Codex Initiative, who many of you are here. So thank you for being here, all of you. And of course, a number of the activities are dedicated to you. And I think actually without further ado, we're going to be ready to start the first session.
We share with pride for a world where health is on our side. Collaboration and science as pillars. G-Force, we aim for rigor. G-Force, together we thrive. Health and safety will always strive. From to forth, let's make it right. G-Force, for safe food we fight.
So The first session will be dedicated to the Arab section of AOAC International. I would like to invite Dr. Amina Souf, Research Manager in the Global Food Regulatory Science Society, and also President of the Arab section of AOAC International, to chair the session. Thanks, Amin.
Good afternoon, everyone. Thank you, Professor Sam. So, thank you for being with us here this afternoon for this first session out of several sessions organized in the framework of our Global Food Regulatory Science Symposium for this year, collaborating with the Dubai International Food Safety Conference. So, once more, thank you so much for being present with us. The first session for this afternoon is titled Performance Criteria for Acceptance of Quantitative Gluten Methods, Development of Arab Analytical Method Standards for the Determination of Gluten in Gluten-Free Food. So for this session, we will be joined by 4 speakers presenting 4 very interesting presentations, starting with Professor Samuel Godefroy, who will introduce to us this issue of food allergens and gluten management from a regulatory perspective. Then we will be honored to have a presentation, a pre-recorded presentation from Dr. Laura Allred, from a co-chair of the Gluten and Food Allergens program at the AOAC International. And then we will have a presentation by our technology provider and sponsor, RBioPharm, Dr. Stefan Schmidt, presenting the performance of ELISA-based analytical methods supporting this compliance effort with gluten-free regulations. And then we will have the chance to discuss a bit our initiative for the region, titled the Arab Gluten and Food Allergen Management Initiative. We already started and launched the activities of this initiative, so we will brief you about our achievements, about our perspectives, short-term and long-term perspectives. And finally, Professor Samuel will be moderating a round table discussion, discussing all together how we can boost forward the adoption and development of Arab standard for the analytical performance criteria for gluten testing methods, particularly for food commodities of interest for the Arab region. So thank you once again. And here I would like to invite Professor Samuel to the podium. Professor Samuel will launch our discussion for this afternoon with a presentation, as I mentioned, titled Introduction to Food Allergens and Gluten Management from a Regulatory Perspective. So thank you, Professor Samuel, and the floor is yours.
Thanks very much, Amin. And I guess actually this presentation is really aimed at setting the scene. When we are developing analytical methods or standardizing analytical methods, it's always important to see why we are doing that. And we are doing that generally because there is a public health issue that needs to be managed, but also there are some regulatory requirements that are associated with that. And many of you may have heard me actually give this type of talk or a similar type of talk, but it's always important actually to set the scene in this particular context. So I'm going to see if actually okay, it was not on, so at least. Okay, so in setting the scene as well, I will be focusing quite a bit on the international context as set by the Codex Alimentarius Commission, including the work that's going to be discussed, in fact, next week as part of the 48th session of the Codex Committee on Food Labeling. Now, I believe that you don't need convincing that food allergies are considered to be a public health issue, and food allergy incidents can have important repercussions, including the loss of life when basically an incident leads to anaphylaxis. We know at the same time that these food allergy incidents are preventable. And the way they are preventable is by making sure that the person who is allergic to a particular food knows about the composition of the food such that they can avoid that food. We have seen an increased interest at the international level about how this issue can be managed. Celiac disease is another issue. So this is an immune-mediated condition It is not an allergy. It is some form of intolerance, but it is basically immune mediated in the sense that there is a recognition of the particular ingredient or the culprit ingredient by the individual that has this condition. Generally, 1% of the population worldwide is affected by this condition. It tends to be also, there is a genetic predisposition to this. Research is still underway in order to know how the phenotype actually is expressed in individuals. And there is a particularity of this condition in that the symptoms of this condition, which are essentially gastrointestinal symptoms primarily, so essentially cramps and aches, actually these symptoms tend to be quite similar to other conditions. And that's what makes celiac disease late in the diagnosis. Unfortunately, when not diagnosed early enough, and when gluten is not avoided early enough, the situation can deteriorate, mainly by malabsorption of nutrients by these individuals, because basically the villi of the intestine gets deteriorated, and it can lead even to conditions such as osteoporosis and even cancer. Now, there are some anecdotal data related to celiac disease in this region, in the Arab region, that the prevalence seems to be higher than the 1% that was reported internationally, and this is still subject to research. Now, for those two conditions, beyond the public health manifestations of these conditions, meaning the medical costs, etc., there are other repercussions, and they are mostly social repercussions. I don't know if any of you has invited to your home a person either with celiac disease or with allergy. And actually, the crisis that you will be living is what am I going to be feeding that person? And am I going to be causing a particular issue to that person? Well, imagine actually this in a social setting and the impacts that that can have in daycares, in schools. When in a classroom, there is one child that is affected with food allergy, basically the rest of the children have to follow practically the same diet, in the sense that if peanut is the allergenic culprit, all the kids cannot bring any peanut to the school. And of course, that can cause some social repercussions because the person may feel ostracized and excluded from the community. So there are also psychological impacts that are associated with these conditions. Now, unfortunately, until now, and while research continues, there is no cure to neither celiac disease nor food allergies. So the only way we can do this is essentially to prevent these incidents from happening. How can we prevent these incidents from happening? By making sure people avoid the food. So avoidance is the only risk management strategy that is available. For gluten in particular, it's a long life avoidance. So essentially, you have to exclude gluten from your diet during the entire life. And for gluten, given that we're going to be focusing on gluten for the rest of our discussions here, you know that gluten is actually present in more than the common food that we know. So of course, gluten is present in bread, in cereal-derived products, etc. But in many processed foods, gluten and some extracts of cereals can be included in the formulation of these processed foods. So that avoidance may not be as simple and as easy as we may think. And again, you know, the main element to help consumers with this condition. Now, when you are in a setting, when you are in a food service establishment, you have a direct contact with the producer of the food, with the one who is preparing the food, and you can ask the question, and hopefully they know the answer, okay? But when you are talking about prepackaged food, the only link that you have as a consumer with those who prepared the food is the label. So labeling has been really at the heart of every risk management strategy. And in fact, in order to help this avoidance mechanism to be implemented, we need to make sure that labels are accurate, and we need also to make sure that consumers understand the labels such that they can take action from a risk management standpoint to avoid those food products. And this is basically the objectives of any type of labeling policy that a food regulator will need to promote. Basically, we try to make sure that labels are accurate, so you don't have any hidden source of the allergen or the gluten source. They need to be clear. Basically, you cannot be shopping with a dictionary, particularly for allergens, like knowing that you cannot have beta-lactoglobulin or ovalbumin, because you need to be a scientist to know that beta-lactoglobulin means a protein from milk and ovalbumin means a protein from egg. You need to be able to understand that much more easily. And you should not have a doubt on the composition of your food. So essentially, this policy is what guided every regulator in the way they have developed their labeling requirements for allergen and gluten sources. And these elements can be indeed regulated. Now, of course, when you are a regulator, your primary source is the International Food Standard Setting Body, and that is the Codex Alimentarius Commission. In fact, Codex has led the way in the development of these requirements, and we have witnessed the first Codex standard in this regard in 1999. That was the first standard about the labeling of allergens. We have witnessed standards about gluten earlier than that in the late 1980s, where essentially Codex defined what a gluten-free food is and how it should be defined, primarily vis-a-vis the presence of gluten. Now, when you look at what Codex did with allergens in particular, the principle is very simple. Codex says that when an allergen is present, it needs to be declared on a mandatory basis in the food, and it needs to be declared in a simple way. So essentially, these are the situations that we discussed that we need to avoid. You cannot have something that leaves you a doubt, flour. You cannot have something that you cannot understand, ovalbumin. And if you have mixes that may contain, like seasoning, that may contain, I shouldn't say the word may contain, because may contain is another thing, but that may have in their composition an ingredient, so in this case, deliberately added, that have an allergenic potential, basically that has to be disclosed. So primarily, this is what essentially Codex advocated, is that when an allergen is added deliberately in the composition of the food, it has to be declared on a mandatory basis with the common name of the allergen. So essentially, you know that ovalbumin is egg, flour may contain wheat, and in fact, when you have a seasoning, for example, you have to indicate what, if any, allergenic sources may be present. Now, of course, the problem is that we have applied the labeling of ingredient for a number of years, and that has been quite successful. And in fact, after the adoption of the standard in 1999 by Codex, most of the international food regulatory jurisdictions followed suit, and we have basically allergen labeling requirements across the world, including in our region here. The problem that we have witnessed, however, is that there is another labeling that was not covered by a Codex provision. And that other labeling is not the labeling of ingredient, and that's the labeling of unintended presence. And you see here the situation where you have a very simple product. By the way, we picked this product in Quebec, in Canada. So you have a product that contains chicken and salt. And when you look at the may contain statement, the may contain statement contains basically eggs, nuts, sesame, sulfites, and seafood. You have more in the may contain statement than actually the composition of the food, which tells you that there is a problem in the way this may contain statement has been working. And in fact, that's essentially the situation that we have seen over time, is that the frequency of the precautionary labeling, so the frequency of use, will actually result in more incidents. Why? Because basically people stop paying attention to that, and they will say, well, they're putting that just to protect themselves. So essentially, I'm going to take the risk. And basically, they will eat a product, and as long as they don't have an incident for the first time they have it, they will do it a second time, third time, until unfortunately something happens. And that's essentially what now the new work of Codex tried to do. So from 2018 onward, Codex, after close to 10 years of no work on allergens, decided to reopen the work on allergens. And the work was opened primarily by developing guidance to food industry and under the auspices of the Codex Committee on Food Hygiene in the form of a code of practice on allergen management for food business operators. Basically, this was quite a breakthrough to try to improve the practices of food allergen management by food producers. Basically, we tried to have a harmonized and proactive approach in the way allergens are to be managed, mainly to look at how to identify situations of cross-contact, how to identify situations of undeclared allergen, and to put in place the control measures. Basically, you're adapting your good manufacturing practices, your good hygiene practices to a hazard that is now a food allergen. Now, I'm not going to go in details on this because otherwise I will take a lot of time, and I would like to zero in on gluten and on methodologies. But really what I meant to do here through this overview is just to bring you up to speed on the latest development internationally, and particularly what is happening at the level of the Codex Alimentarius Commission, particularly that this is going to be discussed, in fact, next week in terms of continuation of improvement of the conditions of labeling of allergens. So, of course, one of the things that we were faced with, particularly to try to manage the issue of food allergens, is whether we have up-to-date scientific information. And that's why CCFL, so the Codex Committee on Food Labeling, but also CCFH, the Codex Committee on Food Hygiene, resorted to an expert consultation and asked specific questions that help them update the risk management approaches for food allergens. Basically, the questions were three questions. First of all, we had a list of food that we considered as priority allergens that we identified in 1999. The question is this list still up to date? Do we need to update it? And what is the scientific foundation that we should rely upon to consider an allergen a priority allergen? Because, you know, any protein-containing food has the potential to be an allergen. Why do I call this a priority allergen versus another one? So that's question number one. Question number two, we all know that allergic reactions are threshold-based reactions. What do we mean by that? Meaning that there is a level, a threshold, below which people would not have a reaction. Is it possible to identify this threshold? Because we could use that for the purposes of risk management, and particularly for the purposes, and this will address the third question, is there a scientific foundation, particularly in the form of a risk assessment, that we can use on the basis of which we can determine whether or not a precautionary allergen labeling is needed? The reason for that is to make sure that I will not use this allergen precautionary labeling unless I actually need it. And that's essentially what happened. Basically, we had about 2 years of work of an expert group. They generated, in fact, there is a fifth report that was generated. So basically 5 reports were made available and attempted to answer these questions. And in fact, some of the outputs that I'm sharing very quickly with you is that the list of priority allergens was indeed updated. For example, soy was considered no longer to be an allergen of global relevance, whereas sesame was considered to be an allergen of global relevance. The second element that actually the expert consultation advised about is that there may be allergens that are of regional relevance. The examples would be celery, mustard, lupin, these could be actually allergens of regional relevance. And also, what the expert group did is they developed criteria related to the when do I consider something as a priority allergen? And these criteria actually accounted for three elements: the prevalence of the allergy, the severity of the impact, and the potency. The potency meaning that a low level will induce an impact. So those were the three criteria on the basis of which an allergen was to be deemed priority allergen. And also, we were able to have this answer to the threshold question. And the answer to the threshold question was relying on an assumption that was made by the expert group, basically that a level of protection of 95%, meaning if I take 95% of the population not reacting to something, the level that I can identify for that would be protective enough. So basically, they used what we call the ED5. So it's a level of allergen expressed in milligrams of protein, for which basically 95% of the population will not react. And they consider that those who reacted at this level or lower than this level, the reactions are likely to be mild reactions. They will not be necessarily acute, effects. And finally, what the expert group did, basically they developed guidance on a risk assessment-driven approach that will indicate whether or not a precautionary allergen labeling is needed. And in fact, right now, this type of advice is going to be used during the discussions that are going to be taking place next week in Quebec City when the Codex Committee on Food Labeling meets during its 48th session. And we'll see, hopefully, some advancement in the way precautionary allergen labeling are to be managed. So as I mentioned, over the last 20 years, in fact, we've witnessed quite a bit of improvement in the way allergens are managed and are regulated with the adoptions of many countries of the guidance that was developed by Codex. And the expectation is that this will continue with the adoptions of the new provisions, hopefully, that may be recommended next week. For gluten now, we already knew that gluten is a threshold-based condition. Now I'm talking about celiac disease. The celiac disease-based reaction is also threshold-based. And here, we're not talking about situation and immune-mediated reaction that would lead to anaphylaxis. We're talking here more about the impacts on a chronic basis. So you may consume a product. It may have already an impact on you with an acute manifestation that is not life-threatening, thankfully. So it's not actually leading to anaphylaxis. It is, however, discomforting. It's not something that people actually like. The problem, though, is that the chronic exposure can also lead to manifestations, as we mentioned, that can be malnutrition, osteoporosis, and cancer. And there was also the identification that there is a proportion of celiac individuals that may have what we call silent celiac. So essentially, they have the condition, but the only way they know about that is essentially when they do an endoscopy. They would not necessarily feel those acute symptoms that alert them to the situation. An attempt was made to qualify the threshold for gluten, and it was found that 10 milligrams of gluten per day, so per day when you consume that, for the majority of celiac individuals, this will not lead to a health impact or a detrimental health impact. So, of course, this is still requiring further research because this is an area that continues to be developed. Now, the other Codex committee that worked on gluten is the Codex Committee on Nutrition and Food for Special Dietary Uses. Because again, for individuals that have celiac disease, they need to seek food products that are specific to them. Gluten-free foods are foods for special dietary uses. And what CCNFSDU did was to try to develop conditions for what we call a gluten-free food. And the condition that was identified well, moved over time with the levels and with the science. So right now, we have identified under the Codex guidance that a food that contains 20 parts per million or less in protein of gluten sources would be considered to be safe for celiac individuals for the majority of those, and therefore gluten-free foods can be defined with this 20 ppm threshold. As a result, the standard that was adopted, in fact, I said in the 80s, it was in the late 70s that the standard was developed, was amended in order to actually include this provision of 20 parts per million. And you see that this 20 parts per million threshold is included in the Codex standard. So I'm saying this why? Because again, in the context of testing methodologies, you need to target levels in order to know what are the performance criteria that you need to abide by. Now, Codex did not stop at that, but Codex also defined what they consider to be an acceptable methodology for the detection of gluten or for the determination of that 20 parts per million. And in fact, in the Codex standards on methods of analysis, you have essentially a determination of what that method is. Basically, there is an indication that this has to be an antibody-based assay. So essentially, it's an ELISA, enzyme-linked immunosorbent assay, using a particular type of antibody, and it was defined that it is the R5 antibody. R5Y, it's a pentapeptide that is common in all the gluten sources, at the exception of oats. So it's present in wheat, rye, and barley, and they're hybrids, okay? And this is what is being targeted by this assay. Now, the problem is that the assays that were developed for the most part, they tend to be sandwich assays, and they are good to function when the proteins are not hydrolyzed. But when you have a medium where the proteins are highly hydrolyzed, the methodology of a sandwich assay doesn't work anymore. And you need to have another technology or another type of techniques. It's a competitive type of assay that will be able to actually capture those particular samples. Now, at AOAC International, of course, that's what we specialize in, which is the development of standards for methods. And what AOAC International relies upon is two types of methods, either what we call the official methods of analysis, which are essentially methods that have been tested according to interlaboratory studies that take at least 8 laboratories. And you have also another type of methods that have been also checked for their performance, and we call them the performance tested methods, the PTM methods. And those, they tend to have been checked by one laboratory. And of course, when you are talking about methods, you need to make sure that you have validation data that help you make sure that the method that you are using is fit for purpose, meaning for the type of matrices that you are targeting. So in fact, to achieve that consumer safety, we need to make sure that we have the standardized methodologies, but we also need to have this to support the food production processes that are potentially even controlled with these methodologies. So that's what I wanted to share briefly, well, more than briefly, I guess, with you, at least setting the stage on this particular topic. And hopefully, now that you know the regulatory requirements, we're going to be zeroing in on gluten in particular and on the region to see what can be done in order to help us standardize analytical methods for gluten determination. Thank you very much for your attention.
Thank you so much, Professor Samuel. And once again, I would like to welcome all our attendees today during the session and remind you that the session is organized by G-Force as part of the 18th Dubai Food Safety International Conference, DIFSC 2024, and is supported by the Arab section of AOAC International. And here I would like also to welcome our executive committee members, the executive committee of the Arab section of AOAC, who are present with us today. So thank you also for your presence with us and for all the support you are giving to all activities undertaken by the Arab section of AOAC International. Thank you. And just briefly, the Arab section is a regional section of AOAC International, covering all Arab countries affiliated to the League of Arab States. So the section was relatively recently established back in March 2022. So since 2 years, a bit and a half now. And it's trying to serve the analytical community in all Arab countries. We will be taking some questions and doing a lot of discussions and debate, but please note your questions and we will take all the questions during the final session. We will have a panel and a roundtable discussion. And here also I would like to welcome our online participants and ask them also to prepare their questions and to ask them whether using the Q&A chat box in the webinar or even orally and publicly at our panel discussion. Also, I would like to mention that we have an interpretation to Arabic. So all our attendees online can benefit from this interpretation to Arabic. And also our participants here, but you should connect to our Zoom webinar. And then you may have access to the Arabic interpretation. So now we will be moving to our second session. And as Professor Samuel mentioned, let's start by discussing some analytical techniques, some analytical aspects of gluten analysis. And here to launch this debate on analytical techniques, we are honored to have with us, through a pre-recorded presentation, Dr. Laura Allred. Dr. Laura is the regulatory manager at Gluten Free Organization and a co-chair of the Gluten and Food Allergens GFA program at AOAC International. Dr. Laura will present to us all the efforts of the GFA group at AOAC International, setting guidelines for validation of quantitative gluten methods. So we will assist to the pre-recorded presentation of Dr. Laura, and of course, we will have the chance to discuss afterwards.
I'd like to thank Dr. Kasuk and the Symposium for allowing me to provide you with an update on the gluten quantitative method validation guidelines that have been developed by the Gluten Working Group as part of AOAC's Gluten and Food Allergens program. To quickly give my background, I'm Laura Allred. I'm the regulatory manager for the Gluten Free Certification Organization, or GFCO. GFCO is a nonprofit ISO 17065 accredited gluten-free product certifier. And GFCO is operated by the Gluten Intolerance Group, which is a consumer support organization that's been in operation for about 50 years now. GFCO itself is going to be celebrating its 20-year anniversary next year, and it's the largest gluten-free product certifier. We work with over 1,700 companies and over 2,000 brands. And under our certification requirements, we have these companies testing raw materials, finished products, and environmental surfaces. And as such, we have a vested interest in making sure that there are reliable gluten test methods out there. I'd like to start by giving you the short history of the AOAC Gluten and Food Allergens program. The program began at the end of 2020 with thought leader meetings to discuss the gaps in gluten and food allergen analytical methods. The AOAC allergen community has always been a large and active group within AOAC, and there are a number of issues that the group has always wanted to see addressed. Once the gaps were identified, it was decided there were specific needs for both gluten and allergens, so separate working groups were formed, and I agreed to chair the gluten working group, and the allergen working group is chaired by Dr. Melanie Downs from the University of Nebraska. There were a number of gaps that were identified in those early meetings, and one that regularly comes up is the need for reference materials. For gluten, we're glad to have one set of materials that's available for purchase, and that's a set of wheat, rye, and barley materials in oats. The characterization of these has been published, and that reference is given here. And these materials can be purchased through USP. That link is also provided, but they're not certified reference materials, and they only cover an oat matrix. So these are a great resource, but we know there's a lot more work to be done with reference materials. And in the guidance documents that I'll be talking about, we provide some workarounds for the current absence of reference materials in multiple matrices. The group also discussed the fact that there are no non-ELISA confirmatory methods, and that the only guidance currently published is for an ELISA method. And while the group looks forward to developing specific methods performance requirements, SMPRs, for additional methods like mass spec, the panel felt that since antibody-based methods are the ones most commonly used for gluten and allergens, what was really needed now was more specific guidance for the development and validation of antibody-based methods, again, things like lateral flow kits and ELISAs. The Kerner paper that I've referenced here was a great publication in 2013, gave us a lot of the foundation for an updated gluten validation guidance, just like the previous Appendix M, which was based on Michael Abbott's paper, gave a good foundation for the allergen guidance documents that Melanie and the allergen working group put together. While the gluten working group decided to start with generating validation guidance documents for qualitative and quantitative methods, we also have a list of other goals for the group. So as we're wrapping up the review and publication of the validation guidance documents, the gluten and food allergen working groups have actually merged together. And just this past week, we launched a new working group to provide end users guidance, so the end users for these methods. So the validation guidance that I'm going to talk about today is really aimed at method developers, really isn't particularly suitable for labs and manufacturers that are using the methods and may just want to verify their ability to use the kits or to validate new matrices. And we had a lot of end user questions that came up as we were writing the validation guidance, which is why the new working group was formed. So the goals for this new group will be to provide end users with guidance on method verification, matrix extensions, sampling, and data analysis. And we're happy to take new members for this working group. And I'll provide a link where you can sign up for the working group at the end of the presentation. And then after the end user guidance, the gluten working group has additional plans to develop a general SMPR for gluten, as at this time, the only gluten SMPR we have is for gluten and oats, and then to move on to tackle SMPRs for other methods like mass spec. Before we jump into the specifics of the guidelines for quantitative methods, I wanted to mention some of the highlights of the validation guidance for qualitative methods. This guidance is done. We're finished with this, but we're waiting on the publication of one of the references that we used in the document. So now that that's out, the working group is going to be taking one last look at it before it's either published as an AOAC appendix on its own, or it may be merged with the quantitative guidance, which is what was done with the Appendix M for allergens. There wasn't previous validation guidance for qualitative methods, but we were able to pull some general validation guidance from the Kerner paper, things like selectivity panels. And some of the areas where we were able to provide additional guidance were in regards to spike levels for selectivity studies, gluten concentrations for incurred samples used in the matrix studies, and the steps to take when a fractional level isn't found at a low concentration. And this is really helpful for method developers because they often had to perform a large number of tests to try to find a fractional level for the study. Also talks about the study design for the matrix studies, including the number of replicates and the acceptance criteria, and also gives more information on designing and analyzing robustness studies. The qualitative guidance also added a new recommendation for a hook effect study to ensure that the method isn't affected by high concentrations of gluten. And it gives additional details and acceptance criteria for product consistency and stability studies. And that's for methods that are sold as test kits. One important new validation requirement that also appears in the quantitative guidance is the need to use incurred materials for all matrix studies. And given that gluten can come from multiple sources, wheat, rye, barley, and in some countries oats, this guidance and the quantitative guidance both require that all claimed gluten sources be used in the validation study. The qualitative guidance also gives specifics on the study design and data analysis for the method developer study, also known as the single lab validation, as well as for the independent lab study and a collab study. In addition to providing guidance to method developers on adding additional matrices to their validation. So one new term that's defined in the qualitative guidance is the claimed detection capability, or CDC, and that's usually the detection level that's claimed by the method developer. And this concentration must produce a probability of detection of at least 95% in order to substantiate that claim. Like the quantitative document, the qualitative guidance also provides information on sample preparation, and this tells method developers how to use common gluten sources such as wheat, rye, or barley flours from a grocery store, and to determine their gluten content so they can be used as materials in the validation. Moving on now to the quantitative validation guidance. Like the qualitative guidance, this document provides criteria and study designs for the method developer study, again, also sometimes known as the single lab validation, as well as for the independent lab study and the collab study, and this is all with specific details for ELISA methods. These documents were purposely written not to be AOAC specific, but if a method developer were going through AOAC for method validation, the single lab and independent lab studies are required for the AOAC performance tested method program. And with the addition of the collab study, they could be considered for the official methods program. And again, similar to the qualitative document, the quantitative guidance has annexes which contain selectivity panels, sample preparation, matrix categories, and then statistical methods for data analysis. One new criteria that's spelled out in the guidance is the need to have test materials for the independent lab study and for the collab study prepared independently. The preference is that all materials used for this study be prepared by someone other than the method developer, but members of the working group expressed that this can be hard to do, especially if there are a large number of matrices. So the minimum requirement is that at least one incurred material be prepared independently, and then reasoning provided for why all test materials were not prepared independently. The quantitative guidance also adds for performing a curve fit study by running multiple replicates of the standards, calculating the residuals for each replicate, plotting the residuals versus the standard concentration, and acceptance criteria then given for the curve fit and the residuals. For the selectivity study, everybody is used to this covering cross-reactivity and interference, but in the new guidance, we added an additional panel to cover the breadth of the response to different gluten sources, which is basically the number of wheat varieties that are out there. There have been some studies published on differential reactivity of certain test kits to some varieties of wheat, in particular to what are sometimes referred to as ancient wheat varieties. So this additional panel was added to provide the end user with information on how well the kits see these different varieties at levels down near 20 ppm. A sample panel for cross-reactivity is provided, and then the method developer will use this same panel to test for interference at or below 20 ppm. The main part of the guidance then discusses the matrix studies, which are used to calculate repeatability, a new criteria that's called intermediate precision, LOD, LOQ, and recovery. Using one of the study designs that's provided in the guidance, which we'll talk more about a little later. All of these parameters can be assessed by testing 4 levels of gluten, one of which is blank in each claimed matrix. Like the qualitative guidance, the new quantitative guidance also requires that the method be evaluated for how well it quantitates gluten from each claimed gluten source. This table, which is found in the guidance, describes the rotation of gluten sources used in each matrix being claimed. So regardless of the number of matrices that are claimed, the first matrix must be validated using each claimed gluten source. So here that's wheat, barley, and rye. But if the method also claimed oats as a gluten source, that would need to be also tested in that first matrix. And then for each additional matrix, you then rotate the gluten source between wheat, barley, rye, and, if applicable, oats. So the first matrix is always tested with all claimed gluten sources. Additional matrices can be tested with just one gluten source each. The validation guidance also introduces the concept of matrix categories to allow method developers to claim broader food categories and not just individual matrices. This concept is super helpful for end users, as it allows them to apply the method to a wider variety of foods without having to do additional matrix validations on their own. So here we see an example of a matrix category called cereal grains. In order to claim this category, the method developer would need to validate at least 5 matrices, with at least one matrix coming from each of the processing types. So something minimally processed like a flour, a baked item like cakes or cookies, a fried item like maybe a chicken nugget, a pressure-treated item like the pellets that are used to make chips, and then a dehydrated item like breadcrumbs. There are a large number of categories provided in this annex, but none of the categories include fermented or hydrolyzed materials. If a method developer is claiming their method can detect fermented or hydrolyzed materials, perhaps it's a competitive ELISA format, those validations need to be done per matrix and then per the enzyme, chemical, or organism that was used for the fermentation or hydrolysis. So there is no larger category for fermented hydrolyzed foods, and they're not included as part of the listed food categories. So earlier I mentioned a new criteria for the matrix studies, and that's intermediate precision. For most single lab studies, method developers are used to calculating repeatability, which is a variation in results when there's virtually no change in the test conditions. So same kit lot, same day, same operator, same test portion. Intermediate precision provides more real-world information for end users on the variation they can expect when some of these parameters change. So even though there is additional testing required to calculate intermediate precision, the guidance provides study designs like the one that's shown here that allow you to calculate repeatability, intermediate precision, LOD, LOQ, and from one set of data. And if you use a design like the one shown here, where you use 3 separate kit lots in the study, this design can also give you the data you need for your product stability and consistency study. And then the calculations to determine each of these parameters are provided either directly in the guidance or within the additional statistical guidance found in Annex D of the document. Every validation also requires a robustness study, which looks at the performance of the kit when certain steps of the method are varied, such as like extraction times, volumes, incubation times. The parameters to vary are decided by the method developer, or if they're working with AOAC, they'll decide those in conjunction with AOAC. And it's usually based on the common expected errors that an end user might make. So each parameter has to be varied by at least 20%. And Annex D of the document provides study designs and statistical analysis for robustness using the free R statistical package. So as I mentioned, a product consistency and stability study is also required, and guidance is provided for doing this either as part of intermediate precision, like we showed, or as its own standalone study. The guidance also has a long list of information that the method developer must provide, either in the method validation report or in the kit insert. And some important details of this are highlighted. I know this is kind of small. I hope you can see these. They include a requirement that the method developer specify any qualifications or training needed to perform the method. And this is important in the world of gluten because we get a lot of kits that are purchased by consumers. There's also a requirement that all results be expressed in milligrams per kilogram of gluten. This doesn't preclude providing any other results, such as milligram per kilogram of gluten, but the method must describe how to convert all results into gluten. And finally, the method must provide information on the protein source and the gluten fraction or fractions that were used to generate the antibodies. So, did you use barley protein, rye protein, wheat protein? The guidance finishes up by describing requirements for the independent lab study, in which the independent lab will test 1 of every 5 matrices or environmental samples claimed in the single lab validation. If environmental matrices include both environmental surfaces and clean in place solutions, the independent lab should lean towards testing the environmental surface. And then there are similar matrix requirements for the collab study, 1 of every 5 matrices. And per existing AOAC guidance, you have to provide data, usable data, from at least 8 labs for that collab study. And then the guidance describes how to use this data to calculate reproducibility, LOD, and LOQ. And finally, the document provides guidance on how the method developer can add additional matrices to their validation by basically performing additional matrix studies as described under the single lab validation section. As I mentioned, the gluten quantitative validation guidance document is complete, but it's open for public comments. And I've given the link for that here. So if you're interested, if I've said anything here that makes you want to comment on that document, please go in and provide comments on that draft. And I think that's open until November 15th. And then, as I also mentioned earlier, the next project for the gluten working group is our collaboration with the allergens working group on an end user guidance document. And we just, as I said, launched that working group this past week, but we'd also be happy to have additional stakeholders join in. So we're starting out by generating guidance for method verification, basically showing your lab can properly run the method and how end users can validate new matrices in their own lab. So if you're interested in joining the working group, or you know anybody else who might be, the link to join that is given here. So again, thank you so much for the opportunity to provide these updates. And please don't hesitate to contact me if you have any questions.
So we would like to thank Dr. Laura for her participation with us today through a pre-recorded presentation. Just to mention that all presentations will be accessible through our website, gforce.org, with all the material, and all the recordings also, because we're recording the session, will be accessible through our YouTube channel. So please, I invite you to follow our social media platforms and YouTube channel. So as we listened to Dr. Laura's presentation, we discussed some technical terms. So we've been discussing validation criteria, we've been discussing selectivity, specificity, LODs, LOQs, different matrices effect. So, and here our interest, the guideline is mentioning clearly how to extend the validation scope to different matrices. We would be very interested to support the work of extending the validation scope to our matrices of interest for our region, analyzing gluten and gluten-free food. That's it. So now, after assisting to this presentation on the AOAC guidance for validating analytical methods, we are also so glad to have with us Dr. Stefan Schmidt, product manager allergens at ArBioPharm. Dr. Stefan is joining us from Germany, I believe. So, Dr. Stefan, can you hear us? So please, if you're connected with us, if you could unmute yourself. Dr. Stefan will be sharing with us a presentation on performance of ELISA-based analytical methods supporting compliance with the gluten-free regulations. Dr. Stefan? So as also we know that in the Codex standard, as Professor Samuel introduced, these ELISA-based sandwich approaches are mentioned in the Codex standard as suitable analytical techniques to analyze the gluten and to attest gluten levels below or above 20 milligram per kilogram. Dr. Stefan?
Yes, hello. Can you hear
Hello.
me?
Yes, we can hear you perfectly well.
Okay, I had some difficulties with my technical adjustments here, but
Yeah, no
now I
worries.
think it works.
No worries, Dr. Stefan. We can hear you and see you very well. So once again, thank you so much for being with us today. Dr. Stefan is joining from Germany, right?
Yes.
And Dr. Stefan, whenever you like, the floor is yours.
Okay, thank you. Thank you very much for inviting me to tell you something about gluten analysis, gluten-free food production. I try to share my screen now. Can you see now the presentation?
Yes, we can see it very well. If you could please go to slideshow mode.
Okay. Good.
We're seeing two windows. Yeah, we're seeing the two window option.
Oh.
So,
okay.
because I guess you're connected to two screens.
Yes, that was
So
the
if you
presenter mode.
Yeah, yes, if you could share the second screen.
Yeah, I try so.
Yeah, that's
Can
perfect.
you see now?
Yes,
Yeah, okay,
perfectly
good.
well. Thank you, Dr. Stefan.
Good, great. So I will speak today about performance of ELISA-based method for gluten-free regulations. And the first, I would like to give you, sorry, I have now a problem with my presentation. It does not goes on. Gehst du da mal bitte weg? Ja, ja. Now it works. Sorry for that. So my speak will start with a short overview about gluten analysis and how to guarantee gluten-free products. And for that, I would like to start with a short historical review about gluten analysis in the Codex Alimentarius. If you look at the Codex Alimentarius, you will find two different standards. The one standard is the standard for foods for special dietary use for persons who are intolerant to gluten. It's the standard 118. And the second standard is the collection of methods inside the codex. It is the codex standard 234. These two codex standards give you the rules for gluten-free analysis as it is described in the Codex Alimentarius. Starting with a little bit of the historical review, then we have to go back to the year 2003. In this year, Enrique Mendes published the R5 Mendes ELISA, the sandwich ELISA based on the R5 antibody, in combination with a new cocktail extraction. He had already published the R5 ELISA, but he published a new cocktail solution which was specially developed for the extraction of heat processed samples, because Enrique Mendes has had developed that or had detected that especially heat processed products were insufficiently extracted when you use only ethanol for extraction. So he invented with this publication a new cocktail extraction procedure. In the same year, there was a collaborative study where we from our biofarm took part in with our gliadin assay, which is based on the R5 ELISA, and also in combination with the cocktail extraction by Mendes, which is available at our biofarm under the name Cocktail Patented. This collaborative study and the results of this study then were the reason why in 2006 the Codex Alimentarius endorsed the Mendes method, the combination of R5 ELISA and the cocktail extraction, as a type 1 method. 2 years later, then finally in 2008, this combination, the Mendes method, became type 1 method of the Codex Alimentarius, and at the same time was the Codex Standard 118 revised due to the new results. If you look at the Codex Standard 234, you will find there the original publication from Enrique Mendes in 2003 with the cocktail extraction, which is then there described as the type 1 method of the Codex Alimentarius. Type 1 method means it is the defining method. It's like compared to the original meter, the original kilogram. These are the defining or the definitions of the length and the weight. And here, similar to that, we have here the defining method, the Mendes cocktail and the Mendes R5 sandwich ELISA, which define what is 1 milligram per kilogram gluten. As I said, the Codex Standard 118 was revised in parallel, and therein you find the 20 milligram per kilogram limit for gluten-free definition of foods, and described herein also that for the analysis of this gluten concentrations in food, sorry, the R5 Mendes method should be used. So just a short repetition, the Alimentarius type 1 method is a combination of the R5 ELISA, for example, from our biofarm, it's the Reader Screen Gliadin ELISA, and the Mendes cocktail here at our biofarm available at as the cocktail patented. This combination was originally described in the collaborative study from 2003 and became then the Codex Alimentarius Type 1 method. However, we have, if you look at our portfolio, many other methods. Here, just to show you as an overview what is present in our portfolio. In yellow marked 2 new assays that we shortly launched, and with the green mark in the middle, in the center of this slide, you see the Rider-Screen gliadin assay I spoke about before, which is also the AUSC official method of analysis. Since 2021, it has the approval for foods in general, the only method that has this approval available, foods in general. Now you may think, why do we have so many different assays? Because the situations are different. From farm to fork, to use this wording, we have different situations for gluten testing along with the food production chain. When we start at the harvesters, we have the situation to detect gluten, perhaps in oats or in other gluten-free cereals. In this situation, lab is typically not available, and therefore we need a very simple method, and that is the new method that we shortly launched, the first quantitative lateral flow based on an R5 antibody. This goes along with equipment, very small equipment for the analysis of the strips. We call it the smart box. Alternatively, you can use also your Android phone for evaluation. That is a possibility which we provide in parallel with a smart box. More important or most important with this new assay is the data management, because we have here an app on the Android phone which provides you with all information, as you know, also from other computers. However, the most important thing is here the data management via a cloud. With this, in the future, we can provide you a solution also for different sites with a centralized quality management, for example, where your quality managers get from different sites immediately, just in time, the information from strips that tested by different persons. This is just a short overview which should tell you or give you an idea what is possible with data management. On the left side below, you see a table which is already possible nowadays. You can get the information about who is running an assay, when was a sample tested, what kind of sample was tested, who was the supplier of a sample. And so with this, you can do statistics. And in the near future, we can provide you also what you can see on the right side, a colorful dashboard, which gives you immediately overview about what is going on, perhaps in different sites of an organization. This can be also of interest for the intermediate trade, the next step inside the production chain, where the cereals that were harvested were transferred to the food industry. Here, perhaps it's also of interest to get a rapid result if lot is really gluten-free. And with the connection by the data management, you have a traceability of all data from the fields to the final product. However, it is also possible to do this in a lab, and this is perhaps of interest in the food industry regarding quality management. When you have a bigger food manufacturer who has its own lab, he may use the lateral flow for an immediate decision making. When a new lot is coming in, can I use it for my production? But many food manufacturers have also their own lab. And for this, we have also a new assay, the reader screen Easy Gluten. This assay is very efficient regarding sample handling because we have introduced with this new assay buffer tablet, an extraction buffer tablet, which allows for a very simple but sufficient extraction procedure. The assay is also based on the R5 antibody, and in combination with the new extraction tablet, you have a very fast and easy procedure. It can be automated, but in some cases, we also need other assays, which we also have in our portfolio, for example, for hydrolyzed food, which cannot be detected with the sandwich method. Here you need a competitive ELISA format. For this, we also have our assays available. The next situation where you may have or where you may need decision-making analysis is hygiene testing. So inline control production. For this, the quantitative lateral flow can be used, but if you are not interested in quantitative results in this case, there's also a possibility for qualitative method. This is already available since many years in our portfolio. This qualitative quick gliadin assay is especially made for swabbing. The strips here are covered with a foil so that you can use the strip immediately for swabbing. You do not need an additional swab. Of course, in this case, you can also use your lab or a service lab, which provides you with information about your hygiene standards. Most important is that everything is based on the R5 antibody, because the R5 antibody is basis of the official method. The official control laboratories therefore also typically use the Mendes method, the R5 sandwich ELISA in combination with the cocktail patented. This is the type 1 method. Sometimes it is accompanied by PCR method for confirmation, for differentiation if you are interested in the source of the gluten. So is it wheat, rye, or barley? In this case, the PCR method is helpful for differentiation. So here again, just an overview about the different methods. We do not have to look into detail in all these methods. The most important thing is traceability. And this traceability to the Codex Type 1 method is provided by the same immunological target that the R5 antibody detects. If you are in an official lab, if you want to use the Codex Type 1 method, on the left side you see it. It's the combination of an R5 sandwich ELISA, like the Ryder-Skin gliadin, plus the cocktail by Mendez. However, this cocktail solution is not easy to handle. You need to work under a chemical hood because mercaptoethanol is included in this cocktail. It's a long procedure. It takes approximately 1 hour to extract your samples, and therefore some people are asking for easier methods. And for this, we provide, you see it on top, on the right side, the same sandwich ELISA, the Ryder-Skin gliadin, in combination with the cocktail eco, as we call it. It is a cocktail solution which is more convenient, it is faster, it does not contain mercaptoethanol, therefore it is more environmental friendly, and you do not have to work under a chemical hood. Nevertheless, this cocktail solution takes also approximately 1 hour for extraction, and the Ryder-Skin gliadin assay is also from the incubation times above 1 hour. And some people ask for a faster method, and there on the second step, we have here the Ryder-Skin Easy Gluten, the new ELISA method with the extraction tablet, which is a fast ELISA method, 30-minute incubation times, and as I said, a very easy extraction procedure. Then we have, as an alternative, the quantitative lateral flow that I was showing you before. It's because it's a lateral flow method, a very, very rapid method. The results are available in 10 minutes. And with this very easy extraction procedure with the tablet, in all, after 20 minutes, you have your result. Then, last but not least, I show you here on this slide another method that we have developed according to the AUSC SMPR, the standard method program from the AUSC, where they set up requirements for an oat standard method. This is the Ryder-Skin total gluten that we provide here in combination with the cocktail patented. Most important is that all these assays have the same immunological target, and therefore the traceability to the Codex Type 1 method is given. It can be shown, we show it in our validation for the different assays, that the results are comparable for these different methods with the typical or with the original Codex Type 1 method. And therefore the traceability is given. There are other methods in the market which gives you also good results. That's nothing that I would like to tell you here, that other methods give wrong results. But you have a different immunological target, and therefore the traceability to the Type 1 method, the Codex method, must be shown. And in some cases it may work, but in other cases not. And that's the big advantage when the R5 method is the basis of the different assays. What else do we need to regard when we speak about quality management with the gluten analysis? This is the quality management and the quality assurance at laboratories. What can be done for quality assurance in laboratories? We recommend strongly to run standards and samples and duplicate. And this is also mentioned in standards like the CEN standard in Europe, but also in other standards you will find this strong recommendation to run everything in duplicate. Then it is recommended to run control samples with every run for your own safety. This can be commercial available control as we offer here for gliadin, but it can be also your own made control, a spike control, which is based on a typical matrix that you test. What we use here as control material at a biofarm is a cookie. However, you may say, I do not have a cookie that I have to test. I have something else, a different matrix. Then you can also prepare your own control material based on your matrix. That's no question. It is important to run a control at least. What we give you as a manufacturer, along with our validation reports for the different R5 assays, are few criteria for each method. For example, for the Screen gliadin method, we have 6 standards, and you will find in the instructions the criterion that the standard 6, the highest standard, must have an OD value of at least 1.2. If you are below, then the standard curve is not as it should be, and the results become therefore questionable. The lot-specific standard curve we provide you with our certificate of analysis, which is provided with each test kit. However, it is difficult for us as a manufacturer to indicate further criteria, since each lab uses different equipment. Each lab has its own environmental conditions, temperature, humidity is different in different labs in different countries. And of course, each lab has also other people running the test. Therefore, we cannot exactly give you data which are more than the 2 data that we provided here with the standard curve criteria on the COA and the OD value for the standard 6. Here is an example of the standard curve as provided with our COA. We provide you the OD values, and because the OD values, due to the different conditions in the different labs, can be variant of this shown on the COA. Therefore, we recommend the B2B max value criteria. This criterion calculates from the B max value, this OD value from the standard 6, the ratio with the other standard OD values. So the standard 1 in ratio to standard 6 is 5.3%. And independent, if the highest standard is 2.4 or perhaps only 2.0 in the absorbance, all the other values should be lower than in the same ratio. And this can be proven with the B2B max values. So typically, you have a difference between these standard absorbance values and a more or less fixed B2B max value between these standards. Of course, there are slightly different variations from run to run and also from lot to lot, but principally the B2B max value for standard 3, for example, is around 40%. It can be in one case 38, in another case 42%. Due to these variations, we recommend to calculate from at least 10 runs the mean values for the different standards, the standard deviations, and the CV values for these standards. So if you run everything in duplicate, you get mean values and variations from 20 single OD values. And with these, you can calculate your lab internal B2B max values for the standards. And this is your criterion for deciding if a result is correct, if a standard curve is correct, if you can trust in the run that you make perhaps today. The same should be done with the run controls. The control samples that you prepare yourself or the control samples that you get from a supplier. Here also calculate target values from 10 different runs, and then you know if another sample from another run is inside the variation, the typical variation of your control material. So this was my last slide to give you a short overview about what is possible today with gluten analysis and where to be careful when setting up your criteria for quality control and quality management. With this, I thank you very much for your attention.
Thank you so much. A round of applause for Dr. Stefan. Thank you so much, Dr. Stefan, for your participation with us today and for your valuable presentation. We will have, please do prepare your questions if you have any questions, and we will take questions during the roundtable discussion at the end of the session. So we invite you, Dr. Stefan, if you would like to stay with us also for the last session for today's session.
Unfortunately, I'm a little bit in a hurry because I have another meeting today. But please feel free to write down your questions and send me over, and I will answer you then by email.
Thank you so much. Okay. So if any questions, we will, of course, communicate with you. And thank you.
Okay, thank you. Goodbye.
Goodbye. Great. So after those three presentations, we started by introducing the problematic or the issue with Professor Samuel, and then we had a presentation from AOAC International to introduce the different guidelines and requirements to validate our method. Then we had a presentation from our technology provider, RBioPharm, Dr. Stefan, on ELISA-based methods. So now, what we've been doing in the Arab region as Arab section, as G-Force, and as all our stakeholders from the Arab region in order to push further this endeavor of managing gluten in gluten-free food or food consumed by celiac patients. So if you allow me, I will be presenting and sharing with you first a video of several initiatives we've been undertaking in the Arab region, doing hands-on trainings on gluten analysis, using ELISA-based approaches, techniques, supported by our biofarm and by our partners and stakeholders from the Arab region. And here we would like to showcase the Jordan experience. We had the privilege of organizing a training in the military labs for quality control, supported also by our partners in Jordan, the JESMO, the Jordan Standardization and Metrology Organization, and JFDA, the Jordan Food and Drug Administration. So please, let's see the video of one of our successes in the region. So now, what have we been doing in the Arab region under this gluten and allergen management initiative for the Arab world, as launched by G-Force and supported by the Arab section of AOC International? So this Arab gluten and food allergens management initiative for the Arab region, three main objectives. So first, to promote safe food alternatives for celiac individuals, as we've been discussing and speaking since the beginning of the session. Two, to support robust regulatory approaches for gluten-free foods. And three, to develop enablers for evidence-based and data-driven decision-making in this field. So that's very important. As we can see, we need to have data. We need to rely on data. We need to generate data for the region representative of the situation of the region. So that's mainly the three main objectives for this initiative. Now, why this initiative? To answer a few questions. One of these questions to be answered: what is the level of exposure to gluten following a gluten-free diet in Arab countries? So we need to have a question for this answer. We have our celiac community in Arab countries, and we have foods sold as gluten-free, labeled as gluten-free, or are common food consumed by celiac patients. So the idea is to know what is the overall exposure of celiac patients to gluten coming from this gluten-free diet, whether labeled as gluten-free or commonly known and consumed as gluten-free. So that's the first question. In order to answer that, we need to have data. We need to generate data for the region. A second question: is the Codex standard or threshold 20 milligram per kilogram or 20 ppb protective enough for the celiac patients in the Arab region? So in order for us to know, to answer this question, we need also to account for the exposure of our celiac patients to gluten. We need to have an idea about their consumption habit. We need to generate consumption data. And then we will try to apply this threshold and see whether, following their consumption habits and profiles, this threshold is convenient for the Arab region. So that was also a second objective and a second very important question to answer. A third question: is there enough supply of alternatives to gluten-containing foods? So now here we're speaking about innovation in this field, alternatives, Do we have enough alternatives as a gluten-free food for celiac patients in the region? Now, in order to start answering these questions, we've been doing several activities, and we've launched several initiatives for now. The first one is to support the Arab network of celiac patients associations. So we have national celiac patients associations, and we have a network and a certain coalition of celiac associations in the Arab world. So we've been trying to support this coalition through several activities, among which we established very good connection and network with the Jordanian Celiac Patients Association, the Egyptian Celiac Patient Association, now the Omani Celiac Patient also Association and activities. And we've been trying to invite representatives of these celiac associations to participate in similar conferences, panel discussions, in order to make their voices louder, let's say, in the region and heard. Also, we've been trying to organize and facilitate through GeForce and through the Arab section common meetings between Arab associations for celiac patients and international associations, particularly from Canada and the region of Quebec in Canada. And here, so the first slide was all the activities we've been undertaking with the Jordan Celiac Patients Association. And here we can see some pictures of our activities with the Egyptian Celiac Association. The president of the Egyptian Celiac Association participated to a very important international conference, the first Egyptian Global Food Safety Conference and Symposium, which was held in Cairo. The second endeavor or second initiative, in order always to answer those questions, was working towards standardizing analytical methods for gluten testing. Okay, so that's, as we mentioned since the beginning of the session, one of our main objectives. And in order to do that, we've been undertaking several activities. The first one, we launched a call for methods through the Arab section of AOAC International. So the idea here was to try to make contact with Arab laboratories working in gluten testing and trying to see what they are actually doing. How are they analyzing gluten? And here, when we say how, we know that ELISA-based techniques are commonly used. That's for sure. But we need to know more details related to the types of samples they are analyzing, the sample handling, the sample preparation, the extension of scope of validation of ELISA, as Dr. Stefan also mentioned, to very particular and specific food commodities for the Arab region. So we invite you, or if you have contact with any lab working on gluten testing, to answer this call through the arabaoac.org website or 3gforce.org website. And the idea here is try to first understand, scan the Arab region scene related to gluten testing, and afterwards trying to make some harmonization and some alignment between the used methodologies. Again, in this effort also to harmonized and reaching a certain standardized approach, we've been undertaking some training, as I mentioned. So till now, we've completed two trainings. A first one in Jordan with the military labs for quality control. A second training with the Omani Food Safety and Quality Center in Muscat in Oman. The idea here is to train lab technicians and lab workers to sandwich ELISA-based techniques for gluten testing, again, in gluten-free food or gluten-free labeled food. And you can see here two pictures also of these activities. Always with this effort, we've been working on building also an online platform This online platform will be accessible to interested parties, laboratories, different stakeholders, and through this platform, they will access different training materials, video recordings for training materials, discussion sessions regarding all different problems we may face when analyzing gluten and gluten-free labeled food. And always the idea is to expand this knowledge of gluten testing to all stakeholders in the Arab region. Once we do this effort, the harmonization will be much easier afterwards. A third initiative to answer the questions, to gather some food consumption data. And here also it is and it will be a very important effort. We need to have food consumption information and data targeting celiac patients and very specific diet, the diet consumed by celiac patients in the Arab region, in order for us to perform risk assessments and dietary exposures to gluten. So we are pushing towards launching this effort, collaborating with the different stakeholders and particularly competent authorities in Arab countries. And of course, at the end, in order to be able to calculate, to evaluate, to estimate some exposures, and trying to answer the questions we stated at the beginning of my presentation. So thank you so much for your attention. That is our initiative. So thank you for giving us a hand of help in any way you can, and thank you so much. Great. So now to finish our session and to summarize everything, please allow me to invite Professor Samuel again to the podium. And Professor Samuel will lead us through a panel discussion and a roundtable, actually, discussion on all these topics in order for us to reach some conclusions for today's session and hopefully some perspectives. Professor Sam, please.
Just going to try to see. Well, thank you very much, Amin, for that. And thank you for those who are staying still with us. We caught up actually with time, thanks to Amin's succinct and comprehensive summary nonetheless. I'm just going to actually now ask you very quickly here in the room, if I ask you, is there a method, a standardized method in the Arab region that is identified by a standardization organization, recognized by a standardization organization to test gluten in gluten-free foods? Is there one, an established method that is recognized by a standardization organization in the Arab region? So for example, I'm sorry, actually, Ustaz Ahmed, I'm gonna turn to you at the GSO level. Based on my understanding, right now you have some gluten methods in the listing in the GSO, but they are mostly to quality gluten methods. This is to test for the gluten content of food. So this is quality gluten content food, but for methods to test gluten in gluten-free foods, my understanding is that there isn't a standardized method, but you can correct me if I'm wrong on that one.
Yes, thank you. We have two standard GSO for test the gluten now, but adopt from
ISO standards, yes.
Yeah.
But I believe that they are quality. They're not for gluten-free foods. These are quality standards for testing of gluten content, which is another actually objective. It's an important one because that's actually for quality testing, but not for gluten-free foods, okay? Which is essentially, again, I mean, we have a codex guidance, we do have one, but we do not have a regulatory requirement in the region right now. There is also another element as well. And I think actually this one, however, it is regulated in the region, more or less, depending on the countries, more or less, because Egypt, I don't think there is a requirement, but not necessarily broadly. And this is, what is the definition of a gluten-free food? I believe here the GSO has a definition, and it is the Codex definition, so which is basically the 20 parts per million cutoff. But for some countries, Egypt comes to mind, I don't believe that that's actually fully regulated at this point. And based on what we are hearing from the anecdotal evidence that is available, first, the fact that celiac disease seems to be more prevalent in the Arab region than in other parts, which needs to be verified. This is anecdotal information. A second element as well is that maybe the repercussions are more important than what we think, and that the foods that are available that could serve as alternatives to celiac individuals might be more contaminated than we think. So you see, those are all anecdotal pieces that we need to verify. This may lead us to hypothesize that the cutoff of 20 parts per million that is set by Codex may or may not, and may be the case, suitable for the region. By the way, this would not be the first time that this happens. Australia is another jurisdiction that did not actually apply the 20 parts per million. I believe Australia is considering a 5 parts per million cutoff right now, which is practically a limit of quantification of some of the assays. Okay, so it's almost a zero tolerance, but still with the level that is applied. So the question that is posed, which is essentially what was posed in Dr. Amin's presentation, is the 20 parts per million an acceptable cutoff for the region, for some countries or for the region? And in order to do that, we need to do essentially those studies that we indicated. Those studies, we need to have consumption information for what celiac individuals consume as alternatives. Gluten-free products, yes, but also, because you know, celiac individuals and their families, they generally prepare some of the foods on their own by replacing. So for example, instead of using wheat flour, I'm going to use buckwheat. I'm going to use rice. I'm going to use maize, you know, ingredients. What is the level of contamination of these flours? We need to have an idea about, because again, these flours, particularly these flour products, are likely contaminated with low levels. Why? Because the supply chain is practically the same as the supply chain for wheat, for gluten-containing ingredients. Okay, so we have to estimate what is this level of background contamination. Then we need to know what celiac individuals consume, including what they make out of these alternatives, and to make some simulations with the consumption of gluten-free products, meaning products that make a claim gluten-free, and using these simulations, come up with whether or not we meet that 10 milligram per day threshold, because we have a 10 milligram per day. I mean, it may still be also subject to discussion, but it has the merit of existing. We have a health threshold. So basically, we are hoping to conduct a risk assessment with the data that is to be generated. Now, I can tell you that will take several months, if not actually more than several months, because it will require some investment in the different countries, but we are committed to that. And the commitment that we have is that already colleagues from the Sultanate of Oman are in fact already investing in this particular area. We're hoping that Egypt will do a similar type of investment. And in fact, we're considering that Jordan will be another jurisdiction where that will take place as well. So those are 3 jurisdictions that could be used for that purpose. But to support this, we would like also to come up with a standard or a reference for the methods. And this is the proposal that we are going to be discussing, and we would like to hear your views in this regard. You've heard AOAC. AOAC International has developed performance criteria for gluten methods, and there are already methods that are having an AOAC status, either an OMA status or a PTM status, from several kit manufacturers. The question is, with the changes, first of all, in the gluten method performance criteria, but also with the possibility to adapt these criteria to the region. And when I say adapt, what does that mean? It means that we need to look at what are the matrices of interest that might be more targeted in the region than in the rest of the world. So for example, and this is the example that comes to mind, in our region, we consume far more tahine than we eat in other regions in the world, because we have lots of food that actually are based on tahine. We might be eating more chickpeas, hummus and etc. And that those could be alternatives to gluten-free, or they can constitute an imported source of gluten-free foods. So we need to identify what are the matrices, what are the food types that methods need to perform in beyond what AOAC International may have put on a compulsory basis. This will lead us to refine the performance criteria for the methods and to collaborate with the kit manufacturers. I'm seeing one of them here. And actually colleagues also that may be on the line. So Neogen, our Biofarm, all the producers of the test kits of those proprietary methods, to see, of course, if they are interested in collaborating to refine the method performance criteria, but also to contribute in the generation of the data with these updated criteria. Now, this will be even more valuable if we had standardization organizations, and it's great that we have at least 2 or 3 of those present. So I don't see Nesma anymore, but she was here earlier. So we had GESMO, we have the Egyptian, we have EOS, the Egyptian Organization for Standardization and Quality. We have, of course, the GSO, but maybe others in the region who are willing to embark on this process from the start. The idea is that if we produce the performance criteria and then if we evaluate methods according to these performance criteria, we would like these methods to be recognized afterwards as part of the regulatory processes, whether be it by EOS or the Egyptian Food Safety Authority, the GSO, and others. So my proposal is, in fact, that we constitute a working group with professionals that have some expertise in this area, but also with professionals that are representing authorities that could contribute from the start in the development of the criteria, in the consultation on the criteria, and then subsequently in the collaboration with the test kit manufacturers in order to evaluate the performance of the test kits. So this is the proposal that I'm putting on the table, and I'm interested in hearing your views on this. You see, the objective is what? I'm going to tell you actually because it's always nice to see where do we want to go at the end. Where do we want to go at the end is, I'm a food producer and I want to produce gluten-free foods. I don't want to be actually in the doubt to know which tool I will use for what. I will be going to a particular website, whether be it in Egypt, in the Gulf, in the GCC, in other places, and I will be able to know in ETSMO's website, for example, the Arab standards, and I would be able to know this method was evaluated according to a performance criteria that was recognized by such and such, and therefore is considered to be an official method in that country. And therefore I can use it for that purpose. So if I am a producer, I would like to know that, okay? Same thing also for a test kit manufacturer. If a test kit manufacturer wants to develop new methods that fit the requirements of a particular market, they need to know what those requirements are. And I believe that this can be a good pilot for the work that the Arab section of AOAC International can do, using the general guidance of AOAC International, adapting it to the requirements of the region, and therefore generating some standards for the regions on the analytical side. So happy to hear your views. So the floor is open now. Okay, Engineer Ahmed, please. Oh no. Well, actually, you know what? Given that you have the microphone, how about you give us a view from the GSO side?
No, as IDMO, we are working to harmonize Arab standards, but we have more standards that can use it in Arab regions, but we adopt international standards. Our problem is that in the adoption of international standards, we haven't the capabilities and we haven't experts. And in our region committees, working online, it's not very, I mean, efficiency. So that's why we had one meetings in Saudi Arabia, in Riyadh, and these meetings it was very important because it was physicals, and we had with us GSO, we had with us other organizations from other regions, and I see if we can have support for international of, for example, from GeForce or other organizations to participate with us, to attend with us in this meeting, maybe we can have more standards harmonization between Arab countries. Even if we have, for example, standards, for example, of gluten and other issues very important in our regions. What we need to say that we need support from experts from international organizations, from GeForce, for example. So if we don't have that, Arab regions could not adopt Arab standards at regional level, couldn't adopt international standards at national levels because we have laboratories. So if we have laboratories materials, so support from international organizations is very important for us in the Arab regions.
No, thank you. Thank you very much, Engineer Ahmed, for that. And Engineer Ahmed, by the way, represents the Arab Industrial Development and Standardization and Mining Organization, which is the Arab Standardization Organization. And in fact, is also the host of the Arab section of AOAC International. So in fact, I would say this is exactly what we would like to do, Engineer Ahmed, which is to be able to provide that level of support such that these discussions take place. We know that virtual discussions are always difficult. I mean, unfortunately, some of them will still have to happen, and they tend to drive things. But meetings such as this one, where we are face-to-face, tend to kick off some initiatives. And of course, you know, we benefit greatly from the face-to-face meeting of the experts and the regulators in particular. So in fact, we would like, and that's really one of our ambitions, if organizations sign onto this. And again, you know, we have a number of regulators here. You know, Dr. Rasha represents the Egyptian Food Safety Authority and the specific section in charge of food for special dietary uses. So essentially the section responsible for gluten-free products. We have, as I mentioned, Engineer Nesma from GESMO. We had Hanan. You know, I guess you're not at the same time in the same room. You decide just to alternate, eh? Hanan just quit the room just a few seconds ago, but from the Egyptian Organization of Standardization. So and again, you know, ETSMO and the GSO as the two regional or sub-regional organizations. If we could have your interest confirmed to be part of this initiative, but then I would like to have some signing members who are interested to join this expert group that will review the methods. we will be making a call for experts for that purpose. But if some of you are around the room, are in the room and are interested to contribute to what it is exactly that we want you to contribute to, is basically you've heard the performance requirements of AOAC International as they are being updated, but there are already existing ones. The update is actually not transformative as such. It's more actually an update. It's an enhancement. So the performance criteria already exist. Our intent is to take these performance criteria, to study them, and to adapt them to the requirements of the region, including, by the way, from a linguistic standpoint. That's also another element, to make them accessible in all the languages that are used in the region. English being one of them, but Arabic, and also we know French is also an important language that is used in some parts of the region as well. So our objective is also to make that available in the region, and then to have this group basically look at these criteria and adapt them. But this group will be managed as part of the Arab section of AOAC. So it will be considered as a task team, a technical working group, if you will, of the Arab section of AOAC. Therefore, when the recommendations are made, hopefully we'll look at the governance of the Arab section, so the executive committee of AOAC will be making a determination. But we know that AOAC is only an advisory group. It's not an international standard setting body, or it doesn't have a regulatory power. So this will be even stronger if those recommendations are then potentially, and I'm saying potentially because it depends on your requirements and on your governance, potentially considered for adoption by the regulatory bodies, standardization organizations, and food regulatory authorities. I'm seeing nodding, but yeah, please. Yeah.
From a kit manufacturer point of view as well, as you said, it needs to work for the consumer, but it also needs to be practical. And that's something that we as kit manufacturers can then produce to allow reliable and consistent results. But then also food manufacturers need to be considered with regards to what is practical on their side. Because if the regulators come up with a level far too low to be practically implemented on site with the sort of products that we're talking, where for gluten, for example, it can be very dusty, there's lots of risk of cross-contamination, the same with nuts, then those recommendations can't be implemented. So I think it needs, as you say, cross-section involvement from people on both a practicality, but then also on the clinical and the regulatory side as well.
I think, thank you for that point. So please, first of all, actually, I would like to go through the room for anybody who wants to intervene here. So please.
Yes. My name is Adil Shah. I'm working as a quality manager in House of Pops. So the thing what you were talking about before, like cross-country teams, that is very necessary right now. Why? Let me give you one example. Like if you see ISMA standards, suppose I'm exporting ice creams to Saudi Arabia or Egypt or somewhere else, right? Suppose ISMA will say, like, you have to check 3 parameters, suppose, right? But where you are exporting, that country standard is a little bit different. They are telling 5 parameters. Suppose from Dubai, you are exporting on checking E. coli, salmonella, right? And you are exporting. But their country's standard is totally different, right? They are including this one, but some extra parameters you have to do. So in the same way, if AOAC will lead all this, like Egypt standard, GSO, SFDA, and they made a combined standard, that would be very beneficial for everyone. Like you can export anywhere from any country to GCC, to Egypt, to Lebanon, to Kuwait, Qatar, or from if you are manufacturing either in Dubai or in Saudi or Kuwait, you can give to anyone, right? But right now, current situation is becoming a little bit difficult that you have to follow their country as well, and you have to check from your ISMA standards. So this is my opinion.
Thank you. Thank you for that. Anyone else would like to intervene? I think we have a hand up. Yeah, please, you have a microphone, so that's even better. Sure.
Hi, everyone. Thank you so much. Hala Muhammad, R&D professional. Yeah, I'm looking at this issue from the manufacturing perspective, from industry perspective, where I can see, for example, like misusing of gluten-free products just to be positioned in a higher price and sold as in you know like which is not the norm. I believe like now not only the standardization for this product and the level of the gluten should be allowed in this product, but also from industry perspective, how we can make this like more convenient to the people who really need it, not only as a marketing trend for everyone to be just accessible in the supermarket and to be just manipulated to position the food in a higher price range more than the normal product by just altering, as you said, wheat flour with something like any other ingredient in the recipe. So also about the testing. For me, for example, still I believe this will be more complicated from the industry perspective for the people who produce gluten-free products. And that can definitely impact the price for the end consumer, if that all could be added. How, for example, that could be like managed in a way that it will not impact the cost of the finished product. Thank you.
Thank you. I guess actually, I think you've highlighted an area that is very important to remind us all about, which is in fact the fact that gluten-free foods are considered to be higher value foods, and people actually tend to sell those in a more expensive way, sometimes even double the price than regular foods. So hence actually really the interest to make sure that this sector develops in a nice way, in fairness to the consumer, so essentially making sure that this is not prohibitive to the consumer. And that can happen only by making more alternative available, but also creating more trustworthiness of these products.
But
so
my
really
question
clear
also-
standards.
Sorry, my question also, is it really added value product for the people who does not have any issues with gluten? I don't think so. I
No,
don't
they
believe
they
so.
are added value in the sense for a food producer, for a food manufacturer, it's considered to be a higher value for one reason. They are requiring, if they are to be produced well, meaning if they are to be produced fulfilling the requirements, the international requirements, they need to be produced with a higher level of scrutiny than other foods. Your supply chain needs to be far more controlled because you have an additional hazard here. Your hazard is an ingredient for the other foods. This one is actually considered now to be a product that you need to exclude from your production. And that increased level of oversight that you need to exercise, that extra level of control that needs to take place, can be manifesting by a cost. And as a result, this creates basically the higher value of the product. Now, of course, that higher value, of course, manifests itself by a higher cost to consumers. But what we're trying to do is really to reduce that to the consumer by improving the quality all over the place in the supply chain. If you have more alternatives, if you have more primary ingredients, like you have, a buckwheat flour that you can trust, if you can have a maize flour that you can trust, if you can have tahini that you can trust, because essentially it is produced under clear gluten-free requirements, you may end up, with more availability. We know, I mean, it's a supply and demand type thing. It's with more availability, the cost gets reduced.
But my question also is like, as you said, it's a supply and demand issue. But when we see the marketing of positioning of gluten-free now is becoming as a more healthier product to be for all, even if you don't have a need for that, which is for me is something like there is some kind of manipulation going on in the market.
And that's I think that's another discussion about the way consumer
exactly.
trends,
How to protect
move.
the consumer themselves from being like purchasing something that is very high, expensive to them, and not really like knowing exactly if I really need to use this product or not. You know, like this is my also like
Yeah, and that's why, in
there's a
fact, actually, in
responsibility
this
also.
case, the way we are positioning really the products, it's really products for special dietary uses. So essentially for people who need these products. Of course, if others want to use them, that's their own responsibility. But I think, as you said it rightly, we need to have the appropriate communication about these products. You know, again,
Maybe the channels that this product is being sold at, like, for example, if I am going to the supermarket and I am buying product that I know it's only meant for certain people because of a kind of risk of disease, the communication here is different from if I go and buy it from the pharmacy, in a food section in a pharmacy. I would expect something different from a product that I buy from a supermarket,
Yeah.
from a retail chain.
Yes. No, absolutely. I think actually, you know, we're getting close to the end. If not, we reached the end of the session. But even though I haven't heard really people volunteering at this point, but I'm going to actually conclude with this. For this endeavor to succeed, we need to have a multi-stakeholder collaboration. Some of the stakeholders are already here, and I'm going to actually maybe take them to task, given that they are here, perhaps to sign on their contribution. So we have 3 regulators from the region and two regional organizations. That's actually, for me, the first pillar. The first pillar is the regulators, because we need to have you involved from the start. The second pillar, which is essentially the one that we will need to have. There was a question? Oh, so, Sonia, you wanted to intervene? No, sorry. Okay, I thought we missed you. The second pillar is basically colleagues with expertise in food analytical competency. So, and particularly in ELISA-based methods. And here, actually, we're going to have an open call for the region to recruit those who want to contribute in this particular area. So that's, for me, the second element. I said 3, in fact, it's 4, okay? The third will be the test kit manufacturers, because again, if we're going to be evaluating methods that are proprietary methods, they need to be involved. We need to involve those who produce the method. And the fourth will be the food industry, some representatives of the sector, the producers themselves. I think with those 4 pillars, we are able, if we can have actually a working group that is based with representation from these, with different roles. I think the regulators are there more to oversee, to accompany, to give their advice from the get-go, because when they start the process with us, hopefully they can use the process for their own purposes subsequently. And then, of course, giving us your regulatory expertise and taking or maximizing or leveraging the expertise of the other 3 competencies, the test kit manufacturers, and the food producers, and of course the analytical experts. I'm not forgetting the consumers. The consumers or consumer organizations will be will be made aware of our progress. But there is no need to get them involved in the working group per se at the beginning, unless, of course, they wish to, or if they have experts in a particular area. Of course, we welcome everybody. So this is really the intent, and I'm going to conclude with this. Those of you who are interested to join, we're going to leave that that flip chart there. So perhaps you're shy to self-declare from the get-go. Just write your name over there. And we will consider actually the start of the creation of this expert group to work on the standardization of gluten testing methodologies in gluten-free foods. With that, I'm going to give you back the floor. I mean, if you want to conclude, or shall I conclude? Okay, I think actually this brings us to the end of this session. Thank you to all those who contributed to its setting. Thanks, Amin, for your leadership as part of the Arab section. And thank you for all the participants and the contributors.